Thank you for the introduction. So first of all, I want to thank you for being here, and also I want to thank the organizer giving me the chance to speak here. So today I'm going to talk about how we assess and improve the accuracies of next generation sequencing informatics at Bina. About more than a year ago, we got acquired by Roche Sequencing, and now Bina is 100% owned by Roche Sequencing. So what we are doing at Roche Sequencing is that we are trying to bring together best-in-class technologies to craft an end-to-end solution. So what does that mean? We want to provide an end-to-end solution from sample preparation to clinical applications. For example, we have Biosystem, Nemogen for target enrichment, and we also have Genia for long-read sequencing, and of course we have Bina for bioinformatics and also software platform. And then we have Ariosa, Signature, and Cap Medical for clinical applications. So what do we see the genomic data challenge at Bina? So for data challenge, we actually see a number of things. For example, scalability is a problem because we are getting bigger and bigger data. We are generating more and more data from not only the DNA data, but also other types of data, including RNA and protein, etc. So big data is a problem, and scalability, of course, is a problem because not only we want to share the data in an organization, we are sometimes also want to share with our collaborators and also across organizations. And then data governance is also another problem that we have been seeing. For example, people have been trying to manage the data, but the data is just too big and they're also very dispersed. And then the people also care about privacies and the regulatory compliance, how we can actually manage the data according to the compliances. And then of course, and most importantly, I think, is the accuracies of the platform or the software solution. Because even if you can manage your data successfully, if the data are not correct or if your softwares are not giving you accurate results, then you are not going to get meaningful interpretation. So at Bina, we provide a solution, and we are also developing a solution called Genomic Management Solution, GMS. So GMS basically is a software platform combining two of our existing products, Bina Rave for secondary analysis and Bina AIM for tertiary analysis. So for secondary analysis, if you don't know too much about these terms, basically it means from alignment to variant calling. And for tertiary analysis, it means the annotation, interpretations, and analytics. So our platform not only provides the secondary analysis and the tertiary analysis, but we also provide a number of other things, for example, like what I talked about before, the data governance for your data, for regulatory compliance, for security, and also for storing your data. And we also provide a platform for you to capture your data into our knowledge base for you to get better results later. So today I want to talk more about the science and how we improve the accuracy, so I'm not going into too much details of the software. So I've been saying it's a big data problem and it's a big biomedical data problem, but what does that mean? So last year there was a paper, a very good paper in PLOS Biology from Stevens et al. Talk about how big the data could be and how big the data that we project or anticipate. So as you can see, from this plot, it's actually very overwhelming. Because when you look at the beginning around 2000 or 2001, it was the first draft of the human genome, and at that time I think it was only about we talk about gigabytes of data, and that's pretty much it. And then after that, there was let's say the Venter genome, the Crick Venter genome, and also the James Watson genome, and it was still in the gigabytes or maybe at most a terabyte era. But then after that, Illumina actually started to pick up, and also their short-read sequencing was advancing. Then, people, for example, sequenced the African genome, the first African genome, and then European genome, and also the first Asian genome, whole genome sequencing using Illumina. And ever since then, the data actually has been increasing tremendously. And as you can see, not on this plot, but also on many other plots, from NIH, etc., that you see the data actually growing exponentially. And what they anticipate, and after all these projects like 1,000 genomes and also TCGA, etc., So we anticipate in 10 years, by 2025, we are going to get more than exabyte of data. If you don't have an idea how big that is, it means like 10 to the powers of 18. So we are anticipating more than that. I mean, in the paper, they say it should be about 2 to 40 exabytes of data from genomics. So what does that mean? For example, in astronomy, we anticipate by 2025 there will be only 1 exabyte, and for YouTube there will be only 1 to 2 exabytes of data. That's pretty much it. And for genomics, definitely we are going to be bigger than that. So if the data are going to be that big of the size, then I mean, we can't always reprocess the data. And I mean, if we are going to get like 100,000 of genomes or millions of genomes, I mean, we want to have the most accurate platform to get the data as the time goes. I mean, we don't want to keep reprocessing them, and we don't want to waste money on that, I mean, if the data are not accurate. So that's why Bina actually has spent a lot of effort to improve the accuracies. And also before we improve the accuracy, we thought a lot about how we should measure the accuracies, because if we don't have a good way to tell whether our platform or our solution is accurate, then how can we improve anything? I mean, how can we see the problems of the current solutions? So we have published a lot. We have published a lot in the last year. For example, as you can see here, these are just some examples of our publication. We published matter SV and SV caller, and we also published a somatic-seq, a somatic mutation caller, and then we also have some other publications and applications. For example, VARSIM, which is a simulation and validation framework, and Hugo GoSet, which is a quick mentor GoSet. I will talk a little bit more the details in my subsequent slides about the research that we have done and the results. So VARSIM is a simulation and validation framework that we published in Bioinformatics in the beginning of 2015. The reason why we came up with this idea was that, I mean, we didn't want to just do a random simulation. We believe that the variants in the genome actually are not random. I mean, they have a meaning, and we have to, I mean, we have to find the best way to simulate that if we want to rely on simulation to do assessment on the accuracies. So what we basically did was, I mean, a very simple idea behind. I mean, we don't randomly generate variants. We take the existing variants discovered by other sources, and then we try to synthesize a genome. We try to synthesize a personal genome and make a synthetic genome. And then based on that synthetic genome, we also use a realistic simulator to learn the error profiles from the sequences. And then we use a simulator to simulate the reads from the synthetic genome. And we believe that the reads simulated from the synthetic genome, they should be as close to as possible to the original genome or to a real genome. And based on those, because I mean, we gathered the variants to generate the synthetic genome, and we also synthesize the reads and simulate the reads. So we know the ground truth, so we know how to measure the accuracies. So we went on to test a number of different applications to see how they perform in different settings. For example, we also we simulated up to 58 of short read sequencing to 100 base pairs, and then we compared the results of different applications. Let's say the BWA MEM using haplotype caller to call variants, and also novel align, doing the alignment, and then also with haplotype caller, and also some other callers like free base. As you can see, the results are pretty interesting. That you can see haplotype caller, it does a really good job after certain size of the variants. For example, for bigger indels after 10 base pair, if you use a traditional unified genotyper or even free base, they're not going to get you all the variants. But if you use haplotype caller, because it's a de novo assembly, at least I mean a regional de novo assembly algorithm, so it can give you a better job. You can do a better job in detecting indels for larger sizes. And the diagram below that you can see some comparisons of some more difficult detection, which is structural variation detection. And we compared a number of tools, including breakdancer, a paired and mapping tools, CNV-nator, a read depth caller, and Pindel, a speed read analysis caller. So they all perform very differently, and as you can see, for different sizes of structural variations, they do perform quite differently. So that also gave us some more ideas to build a SV caller based on that phenomenon. Here is actually just to show you the other part of VARSIM. I just said VARSIM actually is a simulation and validation framework. And so we actually did spend a lot of time to build a portal and also to build a web application for you to validate your results. So for example, if you go to GitHub, our web page, you launch web applications, you can submit the results from VARSIM that you generated from VARSIM to our portal or our web applications, and that actually will give you very detailed analysis, which is like somewhat like a QC metric or like a benchmarking to tell you accuracy results, including F1 score, precision, sensitivity, and a number of very useful things for you to see how your tools actually perform. So just relying on simulation, we think, is not enough, even though we said we want it to be realistic as possible, but it's still a simulation. We want to see how it's going to perform, the tools in benchmarking in real data. Then we went on to build a good goal set, a bigger goal set, at least at that moment. At that moment, when we started the development, there was the Genome in a Bottle goal set NA12878, which was very famous, but they didn't have the structural variations. And we saw the problem, and we also saw some other goal set released during the time that we did the research, but still they were not comprehensive enough. Some of them, they had only the SNVs, and some of them, they had only the structural variations, and some of them, even though they have the SV, but the SV are actually not large enough. And we saw the edge of using the vendor, or some people call that HuRef genome, because the HuRef genome basically was sequenced by single sequencing. So as you can imagine, it's a long read sequencing. It goes up to, for example, like 1,000 base pair, and they also did like 100,000 base pair pattern or mate pair mapping. So that actually can help us to build a better map of structural variations for the quick render genome. And we did a multi-platform and also multi-method analysis or confirmation for the SVs and SNVs that either we detect or we gathered from the literatures. At the end, as you can see on your right, the diagram, you can see that we have a very comprehensive spectrum of the variants, from SNV to very large size of SV, like over 100,000 of base pairs. And if you compare to all the other goal sets, like the lines that you see on the diagram, Genome in a Bottle, Platinum Genome, and also the Baylor College, the BGS goal set. So they are actually very good goal set, but as you can see, we are the most comprehensive goal set, and that goal set basically provided us another way to measure the accuracies of the tools and to benchmark the tools that we are using. So we developed the resources. We don't want to put it in a black box, or we don't want to just claim it and then we say everything that you can't understand or you can't test. So we released the data, we released the results and the resources to the general public, because we think this is a very valuable resource for you to benchmark your tools, not only for us ourselves. For example, we put our resources on our GitHub webpage, and also we collaborated with Genome in a Bottle, so we also put up our data on their website for you to download. So it is basically just like another NA12878 genome, but then it provides a more comprehensive assessment for your variants. We talk about the VARSIM simulation and validation framework, and we also talk about the real data goal set from the HuRef genome. And so what did we do actually more after that? Not only we put it on a website for you to download, so recently FDA, they initiated an effort called Precision FDA to build a portal for people to collaborate and share data and run the analysis. So we did an early development with them, and we put up our applications on their website, Precision FDA portal, for people to use to validate their data and also to make assessment of the accuracies of their tools. So we put up the VARSIM application, and we also put up the HuRef goal set for you to do your own validation. So we actually have very good methods to make assessment of our tools or of any existing tool. But then, we spend some time to think about, so what are the gaps? We don't want to reinvent the wheel. We actually want to improve the existing algorithms. So for example, like I presented before on Vosim, we also saw some characteristic of structural variation detection. Some tools are better in some sizes and some tools are better in other sizes. It's just because they are different algorithms, so they are targeting different problems. So we saw that characteristic, so we went on to take the approach we call either an ensemble approach or integrative approach to integrate different orthogonal methods to do a better job on variant calling, because we think the orthogonal methods, they can complement each other. So it's a very simple way, so we combine a number of different methods. For example, a junction mapping algorithm called Brexit, published in Nature Biotech before, and a read pair analysis by Breakdancer, speed read analysis by Pindell, and read step analysis by CNV Nature. So these are all very popular tools for different methods to detect structural variations. So not only we use all these tools to detect the structural variations, we also spend time to develop our own method called it's called some kind of a soft cliff analysis. That means we make use of the soft cliff information in the alignment to try to cause structural variations. The reason why we did that, because we see that the existing tools, they are very good at detecting deletions, but for large insertions, because of the size of the read, so it's actually not able to detect larger size structural variation. So the soft cliff analysis actually would help to detect those sizes, although it might not tell you the actual size, but it will tell you the insertion point where we expect to see an insertion of size actually bigger than 100 base pair. We published our paper in Bioinformatics again, and we also measured the accuracies. As you can see on the diagram on your right, MetaSV basically achieved the best accuracies among all the tools that we have used. So this is just a performance in deletion. We also have some other performance diagrams for other types of SV that I'm not showing here. So MetaSV is a good tool. I mean, it's a very accurate structural variation caller. But what do we do next, and what do we do more than just building a caller? We try to use MetaSV to see whether it can accurately detect structural variations in the real world. For example, just 2 months ago, Gino and Roberto, which the consortium that we collaborated with, they published a paper called SV Classify. They basically built a goal set, further extending the goal set that they published before for structural for SNVs. They included this time the structural variations. We collaborated with them, and we used a MetaSV to validate that goal set. We found that using MetaSV, we can validate up to 99.7% of their structural variations. And we try to use like different level of validation, including somewhat like a true analysis. For example, we try to use MetaSV to detect structural variation in the parents of the subject. And then we compare those structural variations to the structural variations that they published in their goal set to see whether it exists. Because we believe most of the structural variations should be inherited. And we also spend a lot of time in something even more difficult than structural variations, which is somatic mutation detection. It is difficult because the cancer is something very hard to detect in terms of detecting variants. They are, because of their heterogeneity, because of their purity and contaminations and etc. So we, again, took an ensemble approach and tried to see how we can improve the accuracies. We know that, if we combine a lot of different tools, then we must be able to be more sensitive. I think that's a very intuitive approach. But then the question is, how are we going to be more precise? We don't want to generate a lot of false positives. So we thought about using the machine learning approach that maybe we can actually generate a good set of features, and then we build a good model, and based on that model, we just keep on learning, and then we use that the model that we train to classify our somatic mutations to see whether it's a true positive or false positives. So the way that we do, I just described it, it's an ensemble approach combining a number of tools. They are actually very popular and also very good tools. For example, MuTech from the Broad Institute and FrostScan2 from Washington University, Somatic Sniper, John SNV Mix, and Varday. So they both have, they all have their strengths and weaknesses, but combining them together, we actually get very good accuracies or sensitivity. And after our machine learning algorithm, as you can see in the results down there, that we are basically the best. We achieve like more than 90% accuracies compared to all the other tools. And on the right, the right bottom corner, you can see the real data that we also achieved the same accuracies. Among all the tools, we were the best, and also we generated the smallest number of variants. That means we there is a very high chance that we are more precise than the other tools. And we also participated in the ICGC TCGA mutation calling challenge. And in this challenge, the latest stage that we participated, we achieved a very good result. For example, we achieved number 2 in SNV detection for somatic SNV detection. And we were basically, even though we were number 2, we were only like 0.1% less than the first one. And then we were the first one in somatic indel detection, leading by 1.5% of accuracy, which was not easy. So we have done a lot of researchers. So those were just some examples. We also have researchers, for example, in RNA-seq, and we also have collaboration with other consortium. For example, 1,000 Genomes in the last year that they published the last 2 papers, and we still continue to collaborate with different consortium, including ICGC, PCARD, and etc. So I want to thank the whole Bina science team and also everyone in Bina, of course, also Roche Sequencing and all the collaborators contributed to the research. So this is the end of my presentation, and I'm happy to take any question.